Seminar work 15, analysis the results of PCR analysis. The results of the PCR reaction are usually visualized using gel electrophoresis method. Gel electrophoresis is a method in which DNA fragments migrate through a gel matrix by electric current, and the DNA fragments are separated according to size. The standard of DNA ladder DNA marker is usually included so that the size of the fragments in the PCR samples can be determined. By the bands of PCR sample determined by the DNA ladder fragment size. DNA fragments of the same length forms a strip of the gel, which can be seen by eye if the gel is stained with DNA binding dye. For example, a PCR reaction giving 400 base pairs fragments, BP base pair, would look like this on a gel. 100 base pairs, 200 base pairs, 300 base pairs, and 400 base pairs, and the PCR fragments, PCR reaction sample, PCR sample have the 400 base pairs according to the DNA ladder. The main methods for visualization of PCR products: first, staining the amplified DNA product with a chemical dye, such as ethidium bromide, which intercalates between the two doublet chains. And the two second methods of PCR labeling of PCR primers or nucleotides with fluorescent dyes, fluorophores, prior to PCR amplification. The latter method allows you to directly include labels in the PCR product. The most widely used PCR product analysis method is the use of agarose gel electrophoresis, which separates DNA products based on size and the charge. Real-time PCR polymerase chain reaction, real-time PCR. Real-time PCR, also known as quantity polymerase chain reaction, qPCR, quantity PCR, is a laboratory technique of molecular biology based on the polymerase chain reaction. In monitor the amplification of target DNA molecules during PCR in real time. You see at real time, not at its end as in conventional PCR. The results, the amplification of DNA, target DNA, you see at real time in monitor. And real-time PCR can be used quantitatively, quantitative real-time, and semi-quantitatively above, below a certain amount of DNA molecules, semi-quantitative real-time PCR. And at the end of PCR, real-time PCR, which is that picture, melting products at the end of real-time PCR reaction. Two common methods for the detection of PCR product in real-time PCR are non-specific fluorescent dyes that intercalate with any double-stranded DNA, and the sequence-specific DNA probes consisting of oligosaccharides that are labeled with fluorescent reporter, which permits detection only after hybridization of the probe with a complementary sequence. Watch the video real-time PCR about the real-time PCR reaction.
Reassure real-time PCR detection kits. Reassure real-time PCR detection kits allow the specific identification of pathogens in biological samples in all stages of infection. It is based on the real-time PCR technique, an in vitro technique used to amplify specific DNA fragments and detect its accumulation in real time by measuring the increase in the fluorescent signal. To begin the reaction, the temperature is raised to 95 degrees. At this temperature, the double-stranded DNA is separated into single strands. Then, three major steps are repeated during each cycle. First, denaturation to obtain single DNA strands. Second, annealing. The temperature is then lowered to approximately 60 degrees. This allows the primers and the fluorescent probe to bind to your target sequence of interest. And third, extension. Between 60 and 72 degrees, the polymerase extends the primers and cleaves the probe, which generates an increase in the fluorescent signal proportional to the amount of amplified product. After one cycle of PCR, there are twice copies of the target DNA. These steps are repeated through around 45 cycles. When the starting material is RNA, one first additional cycle of reverse transcription is required. The real-time PCR reaction requires the following components: Tag DNA polymerase, reverse transcriptase, primers, probe, DN-TPS, MgCl2, magnesium chloride, buffer, stabilizers, internal control. Reassure real-time PCR detection kit contains the following components: well strips with all the previous reactives in a stabilized format, positive and negative control, rehydration buffer to reconstitute the stabilized product, water RNA SE, DNA SE free, optical caps for sealing wells during thermal cycling. Molecular diagnosis unit consists of 4 areas: nucleic acids extraction, pre-PCR, setup, PCR, amplification, and results interpretation. The first area aims to process potentially infectious samples and subsequent extraction and purification of nucleic acids with the RNA-DNA Reassure extraction kit. We recommend to dilute stool samples before extraction and use the supernatant to perform DNA-RNA extraction. Homogenize the fecal sample, collect a p-size, and transfer to a 2 milliliter vial with 200 microliter of RNA-safe, DNA-safe free water, PBS, or physiological serum. Vortex the sample and centrifuge. Samples are lysed at elevated temperature in the presence of lysis buffer and proteinase K. Transfer the supernatant to the 2.0 milliliter collection tube. Then add lysis buffer and proteinase K and vortex vigorously. Incubate 10 minutes at 65 degrees and then 10 minutes at 95 degrees. After optimal binding conditions are adjusted by the addition of isopropanol, the lysate will be applied onto the mini-spin column and DNA-RNA bonds to the surface of the filter membrane. Add binding buffer and mix by pipetting or vortexing. Incubate the sample at room temperature and transfer the lysate onto the column mini-spin column set. Centrifuge and discard both the filtrate and the RTA collection tube. Then place the column mini-spin column into a new RTA collection tube. Contaminations and enzyme inhibitors are efficiently removed during the following wash steps, while the nucleic acids remain bound to the membrane of the mini-spin column. Add wash buffer 1, centrifuge, and discard. Add wash buffer 2 and centrifuge. Repeat this washing step once. Centrifuge to remove ethanol residues and discard the filtrate and the RTA collection tube. The nucleic acids are finally eluted from the membrane using elution buffer. Place the column mini-spin column in the sterile 1.5 milliliter collection tube. Add Elution buffer preheated at 65 degrees. Incubate at room temperature and centrifuge. Discard the column mini-spin column. Store the DNA, RNA sample at 4 degrees for immediate use or preserve from -20 to -80 degrees for longer periods. The sample is ready for the next area. Take the ViaSure pathogen real-time PCR detection kit and separate the required number of wells, including samples and 2 reserved wells, for the positive and negative controls that must always be included in each assay. If necessary, cut the required number of wells with the scissors and remove the protective aluminium seal. Reconstitute each one of the wells with 15 microliters of the blue vial rehydration buffer. Finally, add 5 microliters of the purple vial negative control in the reserved well for negative control. After this process, move the rehydrated well to the next area. 2 independent processes are made in the third area. First, the positive control rehydration. ViaSure pathogen real-time PCR detection kit contains a positive control that must be rehydrated before using this assay. It is recommended to open and manipulate before starting with the assay. Reconstitute the red vial positive control with 100 microliters of the white vial RNA-safe, DNA-safe, pre-water and mix with the help of the vortex. Store at -20 degrees after resuspension and divide in aliquots to minimize the freeze-thaw cycles. The second process is the addition of the nucleic acids and positive control to the rehydrated wells. Add 5 microliters of extracted DNA, RNA from each sample in the rehydrated wells, with the exception of the 2 well reserved for the positive and negative control. Add 5 microliters of reconstituted red vial positive control in the reserved well for positive control. To finish, divide the required optical caps number and close the wells. After this process, move the plate to the real-time PCR equipment. Place the plate in the thermocycler. Program the thermocycler and start protocol. When finished, analyze the results with the software installed in the equipment. ViaSure provides maximum flexibility and compatibility with leading open system thermocyclers and the latest technology for the stabilization of products. Ready-to-use kits with all the necessary components for the testing. Long-term stability that facilitates transport and route.
The difference between the real-time PCR and the user PCR reaction concluded in real-time PCR, we can see the amplification of DNA molecule in real time in monitor of thermocycler, and in PCR reaction we detect the results of PCR at the end of PCR reaction by using GEL electrophoresis methods. And in real-time PCR, use the fluorescent dyes hybridized with the nucleotides or probes of DNA. And in PCR reaction, use it for detection ethidium bromide dyes. Okay, good. Our lesson finished. Goodbye, students. Goodbye.